Genetic technology is currently used to prevent the passing of disease to offspring, but in the near future, it will allow us to detect and choose certain traits for our children. As the arguments over the ethical and moral differences between the use of genetic technology to eliminate genetic variants and to enhance ourselves prevail, another debate rises. Where should the line be drawn? Pre-implantation genetic diagnosis technology, also known as PGD, provides various patients with the possibility of a successful pregnancy, especially for patients suffering from infertility or patients that are in danger of passing on inherited genetic diseases or abnormalities to their offspring. Since its introduction in 1990, PGD has been used to prevent the birth of children with conditions such as Down syndrome, Tay-Sachs disease, and cystic fibrosis. The PGD procedure is executed as a part of in vitro fertilization to allow access to the embryo in vitro. Medication is provided to stimulate the production of multiple eggs. The eggs are gathered using an ultrasound-guided needle, mixed with the partner's sperm, and placed into an incubator for fertilization and embryo development to the 4 to 12 stage. Through a process known as embryo biopsy, 1 or 2 cells, also known as blastomeres, are then removed from the embryo at this stage. The embryonic cells removed are then observed using fluorescent in situ hybridization for any chromosomal defects. Only the embryos lacking abnormalities are implanted back into the woman's uterus for about 4 to 5 days later, reducing the possibility of genetic disease. The cells obtained from an embryo at this early stage are undifferentiated, meaning that they can develop into any type of cell, such as a red blood cell or a muscle cell. Scientists today are trying to control the maturity of these cells by inserting specific genes into them, as well as manipulating the genes already present through PGD technology. Scientists have created many recipes for the directed differentiation, or the manipulation of stem cell culture conditions to induce differentiation into a particular cell type. In the near future, scientists may be able to accurately direct the differentiation of embryonic stem cells into specific cell types, not only to treat or eliminate diseases, but to design babies with preferential characteristics. A designer baby's genetic makeup has been artificially selected by genetic engineering combined with in vitro fertilization to ensure the presence or absence of particular genes or characteristics. An experiment on mice executed at Princeton University proposes one way in which PGD can be successfully used for trait selection. Geneticists introduced into mouse genomes an additional copy of a gene, NR2B, that codes for one type of glutamate receptor and is known to play a role in the development of the brain. The resulting mice seem to learn faster than other mice and retain information longer. The NR2B gene exists in humans, prompting speculation about performing the same trick on one of us. Over time, this experiment could lead to the application of greater intelligence to humans, as well as various other traits such as eye color, hair color, and height. There are many medical and ethical concerns regarding the use of PGD, whether it is used to prevent the passing of disease or to choose preferential characteristics for an offspring. Misdiagnosis of chromosomal defects is one of the errors and problems related to PGD. Also, by manipulating genes to control their development, other genes may be affected. Many of the traits that we have are associated with multiple genes. The Doogie mice, for instance, acquired a greater sensitivity to pain with their greater intelligence. Scientists are unlikely to determine a single gene responsible for an attribute. Furthermore, traits such as athleticism and intelligence are affected not just by DNA, but by environmental factors that cannot be controlled in a lab. Challengers of PGD suspect that a new form of society may develop. Some propose that the genetically enhanced will gain authority, viewing others as inferior. Scientific American, in their May 2009 issue, questioned, will selection of traits perceived to be desirable end up diminishing variability within the gene pool, the raw material of natural selection? Opponents of PGD and genetic enhancement believe that scientists are essentially killing humans by discarding the defective embryos. The only way to prevent a child with an abnormality is to deny them existence. Opponents also explain that there is no way to decide which diseases or traits are suitable for PGD or genetic enhancement. To determine which ones are suitable is considered a form of discrimination. Many people believe that a person's religion that is different from their own is a form of disease. Technology is rapidly progressing to the extent that science fiction may become a reality. Currently, PGD is used to eliminate the chance of a couple passing on a genetic disorder to their offspring. As scientists discover more information about human development and human DNA, PGD services will soon be available to choose desirable traits for children. As the moral and ethical debates about embryo manipulation for medical and aesthetic purposes linger on, one question still remains: where should the line be drawn?